In the vast library of life, bacteria hold secrets that have revolutionized our world. These microscopic organisms, often overlooked, are the unsung heroes of many scientific breakthroughs. Their simplicity belies a complexity that has fascinated scientists for decades. In the 1980s, scientists studying bacterial DNA stumbled upon something unusual. Amidst their meticulous research, they noticed patterns that didn't quite fit the existing understanding of genetic sequences. They found repeating sequences of genetic code like a genetic stutter. These sequences appeared consistently, raising questions about their purpose and origin. These sequences, dubbed clustered regularly, interspaced short palindromic repeats or CRISPR, seemed insignificant at first. To the untrained eye, they were just another quirk of bacterial DNA. What purpose could they possibly serve in these single-celled organisms? This question puzzled researchers, driving them to dig deeper into the mystery. Little did they know, they had stumbled upon a revolutionary tool. This discovery would soon open doors to new realms of genetic research and biotechnology. These repeating sequences were not random, they were interspersed with unique spacers. Each spacer held a story, a fragment of genetic history. These spacers turned out to be remnants of viral DNA, a record of past infections. It was as if the bacteria had a memory, a way to catalog and remember the viruses that had once attacked them. It became clear that CRISPR wasn't just a quirk of bacterial DNA, it was a sophisticated immune system. This system allowed bacteria to recognize and defend against viral invaders with remarkable precision. Imagine a defense system that could remember every enemy it ever encountered, ready to neutralize future attacks. This is the essence of CRISPR, a biological marvel that has transformed our understanding of genetics. This is the power of CRISPR. It has not only deepened our understanding of bacterial immunity but has also paved the way for groundbreaking advancements in genetic engineering. From curing genetic diseases to creating resilient crops, the potential applications of CRISPR are vast and transformative. As we continue to unlock nature's secrets, who knows what other wonders await discovery? Bacteria, like all living organisms, are locked in a constant battle for survival. Viruses, their relentless enemies, attempt to hijack their cellular machinery to replicate. CRISPR evolved as an ingenious defense mechanism against these viral invaders. When a virus attacks a bacterium, the CRISPR system springs into action. It transcribes the viral DNA stored in the spacers into RNA, a molecular cousin of DNA. This RNA acts as a guide, leading a special enzyme called Cas9 to the invading viral DNA. Cas9, like a pair of molecular scissors, precisely cuts the viral DNA, disabling the virus and protecting the bacterium from infection. This elegant defense system is both remarkably simple and incredibly effective. The discovery of CRISPR's immune function was remarkable in itself, but the true revolution began with Jennifer Doudna and Emmanuelle Charpentier. These two brilliant scientists recognized the potential of CRISPR-Cas9 to transform gene editing. In 2012, they published their groundbreaking research demonstrating how the CRISPR-Cas9 system could be programmed to target and cut any DNA sequence, not just viral DNA. This breakthrough sent shockwaves through the scientific community. Imagine the possibilities, a tool that could precisely edit the code of life itself. Doudna and Charpentier had unlocked a new era in genetic engineering, and their discovery would soon change the world. Section 4. A quantum leap: CRISPR's advantages in gene editing. Before CRISPR, gene editing was a laborious and imprecise process. It was like trying to edit a book with a hammer and chisel. CRISPR offered a revolutionary alternative, a precise programmable tool that could target and edit specific genes with remarkable accuracy. This newfound precision opened up a world of possibilities for treating genetic diseases. Imagine correcting the single-letter misspelling in the 3 billion-letter human genome that causes cystic fibrosis. CRISPR offered the potential to cure inherited diseases that were once considered untreatable. Its ease of use and affordability further democratized gene editing, making it accessible to researchers worldwide. Section 5. CRISPR in action, transforming medicine, agriculture, and beyond. The applications of CRISPR are as diverse as life itself. This revolutionary gene editing technology has opened up new frontiers in science, allowing researchers to make precise changes to the DNA of living organisms. From medicine to agriculture, the potential uses of CRISPR are vast and transformative. In medicine, clinical trials are already underway using CRISPR to treat genetic diseases like sickle cell anemia and cancer. These trials represent the cutting edge of medical science, offering hope to millions of patients around the world. By targeting and correcting faulty genes, CRISPR has the potential to cure diseases that were once thought to be incurable. Scientists envision a future where CRISPR could eradicate inherited diseases altogether. Imagine a world where conditions like cystic fibrosis, Huntington's disease, and muscular dystrophy are a thing of the past. This technology could fundamentally change the way we approach healthcare, shifting the focus from treatment to prevention. Beyond treating diseases, CRISPR holds immense potential for agriculture. By editing the genes of crops, scientists can create plants that are more nutritious, resistant to pests, and able to withstand the challenges of climate change. This could lead to a new era of sustainable farming, where food production is more efficient and environmentally friendly. Imagine crops engineered to be more nutritious, resistant to pests, and able to withstand the challenges of climate change. Farmers could grow more food with fewer resources, reducing the environmental impact of agriculture and helping to feed a growing global population. CRISPR could revolutionize our food systems, making them more sustainable and resilient. By creating crops that can thrive in diverse climates and resist diseases, we can ensure food security for future generations. This technology has the potential to transform the way we grow and consume food, leading to a healthier and more sustainable world. In the fight against malaria, CRISPR has been used to engineer mosquitoes to be resistant to the malaria parasite, offering a potential solution to this deadly disease. By altering the genes of mosquitoes, scientists can reduce the spread of malaria, potentially saving millions of lives each year. This is just one example of how CRISPR can be used to address some of the world's most pressing health challenges. Section 6. The Ethical Frontier, Navigating the Power of CRISPR. With great power comes great responsibility. The ability to edit the very building blocks of life raises profound ethical questions. The potential for unintended consequences, both for individuals and for future generations, is a significant concern. The debate over human germline editing, which involves making changes to reproductive cells that could be passed down to future generations, is particularly contentious. In 2018, a Chinese scientist shocked the world by announcing the birth of the first gene-edited babies. He had used CRISPR to attempt to make the twins resistant to HIV. This act drew widespread condemnation from the scientific community for its ethical breaches and lack of transparency. Section 7. CRISPR's Future, Unlocking the Secrets of Life. The journey of understanding and manipulating the very building blocks of life has been a long and arduous one. From the discovery of the DNA double helix to the advent of gene editing technologies, humanity has always been driven by the quest to unlock the secrets of life. The CRISPR revolution is still in its early days. This groundbreaking technology, which allows for precise editing of the genetic code, has opened up a world of possibilities that were once the stuff of science fiction. Scientists around the globe are working tirelessly to harness its potential. We are only beginning to grasp the full potential of this remarkable technology. The ability to edit genes with such precision could lead to cures for genetic disorders, treatments for cancer, and even the eradication of certain diseases. But the implications go far beyond medicine. Ongoing research is focused on improving the precision and safety of CRISPR, ensuring that the edits made to the genetic code are accurate and do not cause unintended consequences. This is crucial as we move forward in applying this technology to real-world scenarios, as well as exploring its vast potential in fields beyond medicine and agriculture. In agriculture, CRISPR could be used to create crops that are more resistant to pests and diseases, or that have higher nutritional value. This could have a profound impact on food security and nutrition around the world. CRISPR could be used to develop new biofuels, create novel materials, and even engineer bacteria to clean up environmental pollutants. Imagine a world where we can design microorganisms to break down plastic waste or to absorb carbon dioxide from the atmosphere, and even engineer bacteria to clean up environmental pollutants. The potential applications of CRISPR are vast and varied, and we are only scratching the surface of what might be possible. The future of CRISPR is filled with both promise and uncertainty. While the possibilities are exciting, there are also many challenges and risks that must be carefully considered. The ethical implications of gene editing are profound and complex. As we venture further into the uncharted territory of gene editing, we must proceed with caution. The power to alter the genetic code comes with great responsibility, and we must ensure that this technology is used for the benefit of all humanity. We must proceed with caution, guided by ethical considerations and a deep respect for the sanctity of life. The decisions we make today will have far-reaching consequences for future generations, guided by ethical considerations and a deep respect for the sanctity of life. As we navigate the complexities of gene editing, it is essential that we engage in thoughtful and inclusive discussions about the ethical, social, and environmental implications of this technology. CRISPR has given us a powerful tool to shape the future of life on Earth. The potential to cure diseases, improve agriculture, and address environmental challenges is within our grasp. But we must use this tool wisely and responsibly. The choices we make today will determine the legacy of this revolutionary technology. As we stand on the brink of a new era in science and medicine, let us strive to use CRISPR to create a better, healthier, and more sustainable world for all.